When a biological agent infiltrates an environmental matrix, it begins to multiply. To isolate and neutralize that threat, we first have to capture it. However, an improperly collected sample introduces foreign contaminants. This triggers false positive test results, leading to immediate operational shutdowns and significant financial loss from unnecessary downtime. Modern laboratories rely on tools like polymerase chain reaction, or PCR, to sequence microbial DNA. While highly accurate, these systems blindly amplify the genetic material present in the tube. If the point of origin sampling strategy is flawed, the downstream analytical automation cannot provide a valid result. This is Bacillus anthracis, the pathogen responsible for anthrax. It serves as the baseline standard for field contamination threats because of its high resistance to environmental stress. Achieving a valid biological sample requires the execution of 3 tactical phases. Phase 1 is probability-based targeting. Before any physical collection begins, the sampling grid must be mathematically mapped to mirror the target population. Phase 2 is matrix-specific extraction. Technicians must deploy specialized physical tools designed specifically for the recovery of microbes from the air, water, or surface being sampled. Phase 3 is aseptic lockdown. This involves maintaining a rigid defensive perimeter to prevent the cross-contamination kill chain from compromising the collected specimen. Effective biological sampling functions as a mathematical operation deployed in a physical environment, requiring precision well beyond basic laboratory work. Every environmental remediation project requires a precise statistical blueprint before a single surface is swabbed. The primary objective of probability sampling is to minimize selection bias. The goal is to ensure the extracted subset of data accurately reflects the entire population within the environment. Simple random sampling uses random number generators to give every location an equal selection chance. Systematic sampling relies on fixed intervals, but is vulnerable to natural periodicities. Stratified sampling addresses this by dividing populations into distinct subgroups to target specific threat releases. In a physical facility, this means isolating high-risk nodes, like HVAC air intakes or bioreactor connections, and sampling them more frequently than baseline office areas. These probability models are not just theoretical recommendations, they are mandated by federal agencies for post-release surface clearance. The Government Accountability Office has established specific requirements for these operations. Their standards demand calculated sampling strategies to increase statistical confidence in negative contamination results. Rigorous statistical power is the only mechanism capable of dictating concrete, defensible field operations. Once the grid is established, the physical extraction begins. Each environmental matrix, air, water, and surface, requires specific physical extraction forces to maintain microbial viability during collection. Air operations require capturing bioaerosols down to 1 micrometer, using slit impactors to pull particles onto a solid agar medium. Water operations require processing massive volumes, like 1,000 liters, using tangential flow filtration to concentrate the sample. The choice of equipment is also determined by whether the lab is looking for culturable organisms or PCR-based DNA. Impactors and impingers are used for culture, while dry filters and cyclones are sufficient for genetic detection. Surface operations present a unique challenge. Microbes adhere tightly to various materials, requiring specific physical friction to strip them free for recovery. A porous material is strictly necessary to physically strip a thick biofilm out of a metallic crevice or joint. The data supports this requirement. Polyurethane sponges yield approximately 18% fungal recovery, compared to less than 4% for standard flocked swabs. Operational execution requires using 10 by 10 centimeter physical templates. Technicians apply the sponge in strict horizontal, vertical, and diagonal patterns to ensure thorough recovery. Matching the extraction physics to the target matrix is the only way to avoid catastrophic false negatives in the field. With the sample extracted, the operation moves to its final defensive perimeter, securing the specimen. Cross-contamination refers to the unauthorized transfer of microorganisms from personnel, air currents, or surfaces into the pure specimen. Preventing this transfer requires strict adherence to aseptic technique. Technicians must utilize sterile gloves, single-use barriers, and non-porous protective equipment throughout the process. Environmental intrusion is neutralized by permanently locking the pure specimen behind a secure, tamper-evident seal immediately after collection. These physical barriers are supported by unidirectional workflow facility design. Moving from clean to dirty zones prevents high-concentration material from being carried over into testing areas. To verify that field discipline was maintained, analysts deploy a quality assurance and quality control matrix using redundant control blanks. Field blanks are sterile media handled in the environment to prove that the sampling equipment itself did not introduce contamination. Trip blanks are unopened sterile media that travel the full circuit from lab to field and back, verifying the integrity of storage and transit. Reagent blanks are laboratory process checks used to verify the absolute chemical purity of the testing agents. Finally, spiked samples involve adding known target concentrations to a matrix to calculate the exact recovery efficiency of the testing process. The Centers for Disease Control and Prevention maintains stringent institutional biosafety guidelines that dictate the safe handling and transport of these biological specimens. Because a compromised sample yields compromised intelligence, aseptic lockdown is the critical bottleneck for data validity. Architecting a valid biological sample requires a complete end-to-end tactical chain. Representativeness must be mathematically secured within the probability grid. Biological stabilization must be achieved through precise, matrix-specific extraction physics, and absolute data purity must be maintained by aseptic rigor and the QA/QC blank matrix. Advanced downstream analytical automation is entirely useless without upstream field discipline. Compliance at the highest levels of federal environmental oversight requires the execution of these protocols without deviation. Adhering to these principles protects against misdiagnosis, prevents operational downtime, and ultimately secures public health. Envirogyn is the established authority in executing these rigorous protocols for environmental compliance. Apply relentless precision to every sampling protocol. That is how you ensure absolute compliance,